Okay, so next section for chapter 10, section 10.2, which is, I don't know, hold on. DNA synthesis in bacteria involves 5 polymerases as well as other enzymes. So now we're going to start going over what we call the replosome. The replosome is just all the enzymes that we need in order to actually allow for replication of DNA to happen. It is a very, again, efficient, important process. And so you can imagine that a lot of enzymes are needed. And so the DNA replication is, as we talked about already, it has a semi-conservative and bidirectional, and it's a very complex process, and so it involves many enzymes. So this right here is just kind of like an image just showing you a little bit of some of the couple of enzymes that we have in order to make DNA replication occur. So one of the first enzymes that was actually discovered was by Arthur Kornberg, and he was able to isolate from E. coli, so from bacteria, he isolated polymerase 1. This was the first of the DNA enzymes that were involved in DNA replication. He basically mutated some E. coli and he mutated them so that they weren't able to make polymerase 1. And when that happened, essentially the E. coli stopped replicating. So he realized that DNA polymerase 1 was a really important, like at least one of the important enzymes to allow for DNA replication. He called it DNA polymerase 1 just because that's the first one that we discovered. They had 2 requirements. DNA polymerase 1 had 2 requirements. First off, it needed a DNA template. It needed to have one single-stranded DNA that it could read, and then it could add the complementary nucleotides onto it. And then he also needed 4 deoxyribonucleic like triphosphates, so DNTPs. So essentially, you can see here in this image, I'm actually going to take away my video so you can see properly. You can see in this image you have the precursor, the triphosphate. So in this case, it's a cytosine triphosphate, and this is going to be your growing chain. The cytosine triphosphate, 2 of the phosphates, so triphosphate, there's 3, 2 phosphates jump off, and then this phosphate is going to attach to the 3' end. And when this happens, you can see here, there's a continuous, like this 3' end of that nucleotide that you just added is now available for the addition of the next following, the next nucleotide. So this DNA, this is all basically brought through the process of the enzyme DNA polymerase. So it needs a DNA template to read, right? So this is an example, the DNA template, you have T, G, C, A, C, and then DNA polymerase is basically adding these nucleotides one at a time. It adds first, the nucleotide has to present, the incoming nucleotide has to be a triphosphate, and then when it gets added to it, you only have an NMP, so a nucleotide monophosphate. So it's just one phosphate that gets added to the 3' end on the base on top. And then the 2 phosphate over here, it's called a pyrophosphate, they come out, and they actually help bring up, like they basically energize the process to some extent. Okay. Another thing to note, and this is really important, is that DNA polymerase, if you guys can see, only happens on the 3' end, right? The phosphate group of the precursor nucleotide triphosphate, this first phosphate has to be bind through a phosphodiester linkage to this 3' OH end of the base on top. Okay, and so basically you always need a free 3' end, and because of that, polymerase only goes in the 5' to 3' direction. Okay, so if, for example, oh, I was going to put on the video, but there you go. If, for example, this is my DNA strand, right? 5', and then I have the 3' right here. Let's pretend they're separated, and I'm making now my new DNA, okay? And then the big DNA polymerase is going to have to situate itself upon it and actually make the strand. So the DNA polymerase is working in a 3' direction. So this strand right here, it's going to start making the strand in this direction. This is going to be the 5', right? Because it's always anti-parallel. And this is going to be 3'. So it's going towards a 3'. So there's always a 3' OH free group at each nucleotide, and then it just gets added a phosphate, you know, a nucleotide, an NMP gets added every single time to that 3' OH. So for this strand, the polymerase would join up over here and go in this direction because the 3' is here and 5' is there. So we always go in the 3' to 5' direction. Every single DNA polymerase does that. This is a very important thing to remember. So elongation happens in 5' to 3' direction. So since elongation occurs only in the 5' to 3' direction, there has to be an initial short nucleotide strand called a primer, right? You need to have a free 3' end to add a nucleotide to, just like we saw here. There's a free 3' hydroxyl end, right, of the nucleotide on top, this thymine nucleotide right here. In order for you to add cytosine to it, you have to have this end has to be able to make connections with this phosphate right here. So you need to have a free 3' end. So because of that, that means your DNA, okay, needs to have a little bit of a primer, a little bit of like a nucleic acid already basically made towards it. So DNA polymerase can't just, like in my example right here, I basically did like polymerase just jumps on and then starts elongating. That's actually not what happens. Okay, now so DNA polymerase, this is not the full story, right? If these are my 2 strands that I'm going to be replicating. First off, another little strand is made, okay, so that I have a free 3' OH end on this side, okay, and then another little strand is made so I have 3' OH strand on this side. All right. Funnily enough, these little things, these are called primers, and they're made out of RNA. Primers are made out of RNA. Okay, so this is made out of RNA and this is made out of RNA. And so you may be thinking, well, I want to duplicate my DNA and make another DNA. Like I gotta get another DNA strand. And that will eventually occur, as we'll see later on. But for the meantime, we have little primers to allow for DNA polymerase to happen. So once that occurs, then DNA polymerization happens. Then DNA polymerase can actually jump on, right? So it jumps on to that 3' end. We have DNTPs, deoxyribonucleic triphosphates, right? Just floating around, adenine, guanine, cytosine, thymine, whatever you need. And then whichever one's complementary to the strand that the DNA polymerase is reading, which is called the template strand. So this is known as a template. This is known as a template strand. Whatever nucleotides are actually complementary to it, the DNA polymerase is going to add. And it's going to add following the 3' direction. So it's going to add it going this way for this strand and going that way for this strand. Because you see here, this is 3', the 3' is just going to be extending this way. It's always going to be opposite of your template. All right. And so we have another strand will be made in this direction. And then here, the strand is going to be made into that direction. Always following the 3'. Okay, so 2 things that you need to remember right now is that the DNA polymerase works in the 3', into the 5' to 3' direction. So it always attaches nucleotides, right, to the 3' hydroxyl group, right, of one nucleotide. And therefore, because of that, it requires a primer, which is these little green things right here. And primers are usually made out of RNA. By usually, I mean they are made out of RNA. So this primer is synthesized by the enzyme primase. This enzyme it doesn't have any particular things like DNA polymerase. It literally can just latch on and make the primer, right? So it doesn't need a free 3' end. It can start making an RNA chain, so a little primer from scratch, and it adds the RNA nucleotides one at a time. So if you see here, we have that primer, which gives that free 3' end, and then you can the DNA polymerase can continue to elongate. So the primase is this enzyme right here, the one that's making the RNA primer in the first place. Once the RNA primer is synthesized, then DNA polymerase can latch on and elongate at the 3' end. Okay, the primers are usually quite short. They're about 5 to 10 nucleotides long, and it's going to be base paired to the DNA template. And so eventually, towards the end of replication, we're actually going to remove this primer. Okay, so the 3' end of the primer, this right here, serves as the actual starting point for DNA polymerase. Okay. They also found out down the line that there's more than just There's all sorts of DNA polymerases, actually. The bacteria have more than one DNA polymerase. You can imagine DNA replication is a very intense process. It's a very useful process. They always are going to need it. And so there's many enzymes that make it up. So we have DNA polymerase 1, which was the first one that was discovered, 2, 3, 4, and 5. 3 is actually DNA pol 3. I also call it DNA pol. DNA polymerase 3 is actually one of the main ones that E. coli uses. Okay, and so all of these are to an extent responsible for DNA synthesis, or at least something important about DNA synthesis. All of them require, well, DNA pol 1, 2, and 3 require a free 3' OH end. So that means they require a primer. They can only elongate if there's an existing DNA or RNA strand, but they cannot initiate DNA synthesis from scratch. So they require a primer. They all elongate in like towards from the 5' to the 3' direction. That's why they require a free 3' OH end. And they also all have exonuclease activity. And if you see here, the exonuclease activity is backwards. It goes from 3' to 5'. And that's because this is exactly what they do. So in this example right here, okay, my DNA polymerase is going in that direction towards the 3'. But say as it's putting in the nucleotides, it makes a mistake. So it makes a mistake right here, okay? So what it does is it goes backwards. It's able to go backwards. And since it has, so that's when going backwards, it's going back to the 5' end, right? Which is exactly what it says here. The 3' to 5' end, that's where we're going. We're going towards the 5' end. And as it goes to the 5' end, it demonstrates exonuclease activity. So it literally chomps off and eats any of the nucleotides it just added because it knows it made an error. So it's going to eat get rid of those nucleotides it just added, including the one that was erroneous, that was the wrong mutated nucleotide. It's going to get rid of it, and then it's going to restart elongating in the 5' to 3' direction. Okay, so basically they reverse direction. They basically screech to a stop if they stalled. They screech to a stop if they realize that they made a mistake. They go back, they reverse direction, so 3' to 5', and as they reverse direction, they're just basically eating away at the DNA that they just made. One of those nucleotides that they eat away is definitely going to be that mutated nucleotide that they accidentally introduced. And then after a little bit, they're going to go back and start elongating as per usual. So DNA polymerase 1 also has the ability to have exonuclease activity from 5' to 3', which is just the normal direction of the polymerase. And this is because DNA polymerase 1 has the ability to excise primers. So remember primers, these things right here that we need because DNA polymerase has to latch onto something, these things right here. This is made out of RNA. You don't want to have hybrids of your DNA replicates, your two DNA molecules. You don't want it to be 90% DNA and then 5% RNA. That's a no-no. We want it to be 100% RNA. Sorry, 100% DNA. And so what DNA polymerase does, since it has the ability to go from 5' to 3', the exonuclease activity, it literally takes it out, chomps it out, takes it out, eats away at it, and then DNA polymerase can go ahead, polymerase 3 can go ahead or 1 even, can go ahead and fill in the gap with DNA. Okay? And again, we'll see that happen further on. But right now, we're just kind of underlying the specifics of this DNA polymerase, the DNA polymerases. So in bacteria, DNA polymerase 3 is an enzyme responsible for 5' to 3' polymerization. It's very essential. Polymerase 1 is important because it's important for DNA replication, but more for the fact that it removes the primer and then it fills in that gap that the primer, where the primer used to be. And it also has 3' to 5' exonuclease activity, so does DNA polymerase 3. And 2, 4, and 5, they're involved in various aspects of repair of DNA damaged by external forces. So things like UV light or other kinds of mutagens. These kind of DNA polymerases go back in and fix the actual mistakes. So DNA polymerase 3, this is one of your main ones, and it's actually known as a hollow enzyme. And so hollow enzymes remember, it means it's one big, huge complex that's made out of many, many subunits. And it's not going to work unless all the subunits are present. And so DNA polymerase, as you can see in this like image right here, it has a lot of subunits, right? So it contains the core enzymes. Where are they? Oh, here they are. Core enzymes, right? And there's a bunch of subunits that make them up. The alpha, the epsilon, and the theta subunit make it up. It contains the tau protein, which is one that basically connects the core enzymes to what we call the sliding clamp loader and the sliding clamp, right? So we're going to get into basically what the DNA polymerase 3 looks like. So it's a hollow enzyme. Good word. So the core enzyme, this is where we actually have catalytic activity. See these two little green things? Those are the core enzymes, and this is where the core primary activity occurs. And so this activity is possible because of, first off, the alpha subunit. The alpha subunit is the part of the enzyme that's actually responsible for the polymerization or the elongation of the DNA. Okay. The epsilon subunit is necessary for proofreading. So the epsilon subunit figures out, like notices it every time it makes a mistake, and it has that 3' to 5', that reverse direction exonuclease activity. So it eats away the errors, and then the alpha goes ahead and fixes those errors by polymerizing again. And then the theta is just there to kind of hold everything together. It's important for core assembly. Okay, usually a DNA polymerase 3 enzyme has about at least 2 core enzymes, so 1, 2. It could have more, it could have like 3, but usually there's at least 2. The sliding DNA clamp is basically, the sliding DNA clamp is this thing right here, okay? And it looks kind of like a donut shape because this is going to actually go around the DNA template. This ensures that the DNA polymerase 3 enzyme stays near the template. It actually increases something that we call processivity. Processivity of an enzyme, it just means that the enzyme stays and like actually undergoes its process for a long time. So it keeps the enzyme, the DNA polymerase 3, connected to the DNA template longer, therefore increasing the enzyme's yield. If there wasn't this thing right here, the enzyme would continuously lose its template, the DNA it's actually working on, and kind of fall off continuously. It would be a very inefficient process. So the processivity is possible because of the sliding clamp, right? So I don't know if I have a picture right here. Yeah, you can see the sliding clamp, which is this donut shaped thing, how it goes and kind of surrounds this blue DNA template, right? It surrounds this blue DNA template, kind of tethering the RNA polymerase, DNA polymerase 3 to its template DNA. Okay, so processivity reflects the length of DNA synthesized by the enzyme before it dissociates from a template. The more higher processivity a particular DNA polymerase has, the more it just basically stays attached to its DNA. Okay, so and usually there's going to be, one sliding clamp per core enzyme. So we also have a sliding clamp loader. This is exactly what its name suggests. This is something that loads the sliding clamp, that donut-shaped thing, onto the DNA template. And it's composed of different subunits like gamma, delta, delta prime, psi, and chi. Lots of different subunits are going to actually load the sliding clamp, which is actually composed of the beta subunit, onto the DNA template. Okay, so as you can see, many subunits, but they're all necessary to make DNA polymerase 3 work. If we didn't have one of these subunits, it wouldn't be complete. It wouldn't be the complete hollow enzyme, and DNA polymerase would not work. So you can see here this weird pinkish structure with like these weird donuts. That's the DNA polymerase 3. DNA B is actually another protein necessary for unwinding the DNA, which we'll get into in just a second. So this is their DNA polymerase 3. It's got your core enzymes that are actually doing the synthesis thanks to the alpha subunit. It's got the epsilon as well, the epsilon subunit that is able to proofread and excise any kind of extra, like any mistakes that the DNA polymerase accidentally introduced. And they're all next to the template DNA because that sliding clamp is right. It's basically clamping. Oh, there we go. Clamping the DNA template to towards and tethering the DNA polymerase 3 to it. Okay.